Okay, so in metaphase 1, and same thing, we're still in meiosis 1, what we're looking at in metaphase 1, same thing, is that the metaphase starts with an M, we're looking at the middle. Okay, so spindle fibers attach to the centromere of each chromosome. Instead of lining up in a single file as they do in mitosis, chromosomes line up as homologous pairs so that chromosomes of maternal and paternal origin will end up at the opposite poles. So we refer to this as independent assortment. So how do we know if it's meiosis 1 or meiosis 2? Big giveaway is that it's still one cell. So that is very important when we start looking at the Google Forms that I do give you guys, is that the metaphase 1, meiosis 1, we still have one cell. So independent assortment, all this is saying is that when they line up in the middle, they're not lining up for any reason. It's very random. Okay. Anaphase 1. So anaphase 1, same thing as meiosis, away. Okay, they are going to go away from each other. Spindle fibers shorten, pulling homologous chromosomes apart. Sister chromatids remain together so that each new pole has a full set of DNA. Once again, we are still looking at that one cell. That is what's indicating to me that we are still in meiosis 1, so therefore it is anaphase 1. Telophase, homologous chromosomes begin to uncoil, spindle fibers disappear, cytoplasm is divided, and each new cell is haploid because they contain either the maternal or paternal set of the homologous chromosomes. So, same thing, getting ready to split, but we are still looking at one cell. Now, meiosis 2. This is very important, that we talk about interphase happening first, but that only occurs at the beginning of meiosis 1. Interphase does not occur at the beginning of meiosis 2. It's going straight from that site of cytokinesis in meiosis 1 into meiosis 2, straight into prophase. Okay, so as we go on to here, each haploid cell formed after telophase will proceed through cytokinesis to form gametes. Okay, so when we go through meiosis 2, a lot of the same things that are happening in prophase, metaphase, anaphase, they are really going to repeat itself. And this is very important when we look at that Google form when you guys do your assessment for this week, is that, let's look here, prophase 1. Now, prophase 2. When we look here, a little different is that I have one cell and I have two cells. So when we enter meiosis 2, it's a little bit of a different start because I'm now looking at whatever occurs in prophase, but I'm looking at it twice. What we need to look here is that they are crossing over is not going to occur. Okay, so once again, guys, crossing over does not occur in prophase 2. It only occurs in prophase 1. So when I look at metaphase 2, same thing, metaphase middle, that it's going to start to line up on that equator. But why is it metaphase 2 when we look at a picture? Well, I have one cell and I have two cells. And in each of these cells, they are both, all these chromosomes are going to line up on that equator in the middle. Okay? Now, anaphase. When we look here, same thing, A away. But the difference is they're still pulling apart. But in this case, we have two cells. That they are both, okay, here and here, both being pulled away, okay, but that is indicating why it's anaphase 2, not anaphase 1. Telophase, same thing, you have two separate cells. They're looking at about to split to end up into cytokinesis to get that physical splitting. So I know this was a little condensed, but please do, guys, go look at those YouTube videos of the animations. They will really help you kind of visualize this information. Okay, each haploid cell formed after telophase 2 will produce will proceed through cytokinesis to form gametes. So in this case, this is where we get our 4 sperm or 4 eggs in spermatogenesis or oogenesis. Okay, so a little bit more details of spermatogenesis if you'd like to read through it. Same thing with the females. We talked about we get 1 viable egg, but even though we get 4 gametes, only 1 egg is viable. That the rest of these are a polar body that are not viable in oogenesis. Okay, this diagram will kind of go through a little bit more detail about that process if you'd like to go through it.